really a fun presentation for me to make because I get to tell you about all the cool stuff with bacteria and archaea and not just the cool stuff that I've found. So thank you for that opportunity. And my goal for this presentation is to not, tell you everything cool that I know about bacteria. It's to kind of get you to think like a biologist for the day. So as you're following along, think, how would a biologist think? And so the two types of organisms that kind of exist on Earth are the prokaryotes and the eukaryotes. And most of the time, I think all of the time this afternoon, we'll be talking about the prokaryotes, the bacteria and the archaea. And the main differences between these two organisms, and you can see there's actually a lot of similarity. Everything has a plasma membrane to keep it all together and keep all the interesting stuff together and doing interesting things. It has a cytoplasm, which is the lubricant that allows everything to work together. Everything has DNA, but the main difference between the prokaryotes and the eukaryotes is that the DNA in the prokaryotes just kind of bundles together in the middle, in the nucleoid region. Whereas in the eukaryotes, like us, you actually have a nucleus. And this has a lot of implications for further processing and for how RNA works. But again, at the end of the day, it's very similar. And it has the ribosomes. Everything we know of has ribosomes. So, second tree of life for the day, we have the eukaryotes up here and the bacteria and archaea, which are your prokaryotes. Now, within the bacteria, you kind of have the cyanobacteria, and those are your photosynthesizers. And you have the heterotrophic bacteria, and those are the things that eat sugars that are already kicking around. And I'm going to talk quite a bit about this. So just a kind of introduction, like what are bacteria doing? You have gram-positive and gram-negative, and this all has to do with the structure of their cell wall and their reaction to a gram stain, which is a technique that almost every microbiologist has used at some point. And if you're a bacteriologist, you have a favorite, gram-positive, gram-negative, and I'm a gram-positive person. Some are extremophiles, so they like the really extreme conditions. That's probably what we're mainly talking about today. Again, the cyanobacteria photosynthesize, and some can be aerobic and some can be anaerobic, which has gotten lost in translation up here. But the archaea are neither gram-positive or gram-negative. They have a kind of a different structures to their cell walls and don't tend to react the same way to gram stains. A lot of them are extremophiles, and there's a super huge variety of metabolic activities for the archaea. So the prokaryotic life cycle, unlike our own, is very rarely sexual. Sometimes you can get conjugation and transferring things around, but most of the time you just get this binary fusion. And so basically that starts with, you have your DNA, you might have a plasmid or 2 or 3 or 4 kicking around in there. You have a lot of ribosomes, which are doing their thing, carrying out translation, making proteins. And then eventually when the cell is ready to divide, you get DNA making more DNA, it copies itself. The cells start to pull apart, and a little more, and eventually you get this total fusion of 2 different cellular membranes, and then your peptidoglycan cleaves apart, you have 2 cells. And this looks really simple, but this in itself is a process I don't think we have a full understanding of yet, so kind of just gives you a key into how complex this actually is that we still don't understand this. So I'm going to talk a bit about the central dogma too, and we've already heard about that, but I'm going to talk about it in the context of what can we use each of these molecules for to tell us more about biology. So you have DNA replication. And just having DNA kicking around, you can do almost an infinite amount of experiments and learn almost an infinite amount of things from DNA. But some of the things that we're going to hear about this afternoon is 16S sequencing. So we can take the DNA, sequence the 16S gene, which everything has, because everything has a ribosome. I should say that the 16S gene is actually coding for part of the ribosome. And if you sequence this as a population, you can get a really good sense of all the bacteria that are in that area. And this is an incredibly important thing to know, because we can kind of learn about that environment, what kind of life that environment supports. And since we can't culture all bacteria, we can culture about 1%, give or take, sequencing is very important. We can also sequence everything, so not just the ribosome, we can sequence all of the genes in an area. And this gives us a really good idea of not what's going on, but what could potentially go on. So what that environment has the potential for doing metabolically or evolutionarily. So DNA, which we've already heard, can be transcribed into RNA. And if you want to look at the RNA of a population, you can learn slightly different things. So you can look at all of the RNA in an area in something called transcriptomics, and that tells us about what the bugs in that area are actually doing. So what part of the genome is active in a certain area. You can also do something called 16S rRNA. So instead of sequencing the RNA from the DNA, you're going to take the RNA directly and sequence that. So DNA is fairly stable. You can abuse it and use it and get a lot of information out of it. RNA is not. You look at it the wrong way in the lab and it just goes away on you. So it might be the same thing in the environment. So this might tell us what portion of a population is alive, as opposed to what's there or what has ever been there, because DNA is stable. And then you can also, once you've got into translation and you have some protein kicking around, you can also do proteomics. And you can do that in the environment and see what proteins are happening and what's happening metabolically in an area. But you can also do that in the lab and look at what different bacteria are doing metabolically under different conditions that you can grow them in, provided you can grow those bacteria. So we'll probably hear about all these things this afternoon. Okay, so what do bacteria need? And bacteria need a lot. This is dumbed down to a very simple idea, but they need essential elements. So they need carbon, they need iron. I don't think we found anything yet that doesn't need iron. They need nitrogen, things like that. Most things need water. I think everything we know of needs water right now. Energy, of course, they have to get stuff done. And a lot of them have special adaptations to survive. So I'm going to talk about that a little bit. So growth and respiration. Basically, just like us, you need something to eat and you need something to breathe. And I would say that there's unlimited ways of doing this. You could count up all the ways we know about, but we probably haven't scratched the surface. So if you need organic carbon to eat, like we do, you're heterotroph. Or some things can make their own organic carbon, and that's an autotroph. And if you use oxygen, if you breathe oxygen, you're an anaerobe. And if you breathe something other than oxygen, if you breathe oxygen, you're an aerobe. If you breathe something other than oxygen, you're an anaerobe. So you're going to hear a lot of terms probably. This is just an example, and these can get long and confusing, but just kind of break it down to their terms and think about it that way. So a chemoautotroph is something that can fix its own carbon, so it's not eating things. And it's chemo, so it's breathing chemicals. Oh, I've lost all my pictures. That's not good. Anyways, so there was a picture of a kid here eating cereal. And he is a heterotrophic aerobe because he eats his sugar, you know, the nice sugary cereal, and he breathes oxygen. And a lot of bacteria do this too. And after he's done eating his cereal and breathing, he respires CO2, and the sugar, well, you know what happens with that. Now I have some pictures back. So another way, instead of just the basic heterotrophic aerobe, would be anaerobic. So this guy's not using oxygen, and he's making methane. And there are just tons and tons of ways you can do this. And one way is to use hydrogen and CO2, and out of that you get methane and H2O. So very interesting metabolism. But other ways of making methane, you can make it out of acetate, and if you're eating acetate, you're acetotrophic. Or you can make it out of methane thiol, and you could be methane ethyltrophic. And just an interesting bit of trivia there. When you eat asparagus, that's why it stinks when you pee. So you have all these bugs sitting there making methane. When you're thinking as a biologist, you're thinking, well, what could use that methane? And one of the things that could use the methane are the bugs that eat it. So those are the methanotrophs. So some guys are making it, some guys are eating it. And a really nice example of this is actually in the permafrost. So down deep, where you have anaerobic conditions, you have a nice little community of methanogens pumping out the methane. But if you go higher up in the soil level, where you get some oxygen mixed in, you can have a nice little population of methanotrophs eating what the methanogen's made. So another way of thinking like a biologist, what makes it and what can eat it, and you might find them living together. So another way of eating and breathing are photosynthetic autotrophs. So these guys are using the sun to make their own sugars. So they're fixing carbon. So you see the carbon coming in, oxygen coming out, sugars coming out. My slides aren't translating that great, but so extremophiles are basically what we're talking about this afternoon, and they're any organism that's able to, live, maybe not thrive, but survive in conditions that are normally fatal to anything that's not an extremophile. So you're going to hear all kinds of terms about what kind of extremophile these guys actually are. So you can have thermophiles, and these guys live at high temperatures, and it says 45, and it says 122 over here. So high temperature guys. Cryophiles are anything that can survive at -15 for a given length of time. Hypoliths are anything that can live in rocks in cold deserts, so very extreme condition there. And then you can also, combine any of these things to make other ophiles. So a thermoacidophile is an example of when we combine things, and these guys can survive thermophilic and high acid conditions. So another way of, instead of having adaptations, which I'm going to talk about in a minute, is just go on strike. I'm not working anymore until conditions improve. And bacteria do this, and it's called sporulation. So they pack up some of their genetic material, and they quit working until they get better conditions. And this is a nice little process, and it's a great way. Spores can survive almost anything. Spores can survive on Mars. Good way of surviving. So adaptations are a huge field in extremophile biology. What kind of traits allow you to survive? And identifying these, figuring out how they work, and figuring out what all they can do is a huge part of this field. So I'm just going to talk about this for a little bit. So temperature extremes. I can't obviously go through all of the adaptations to extreme environments. So I'm going to hit on thermophiles and psychrophiles, and we can kind of compare and contrast these two. So thermophiles are things that are surviving up here. And you can see if everything grew and divided at the same rate, extrapolate it from the Arrhenius plot that you'd expect based on the things that are kind of in the middle, you could see they'd be growing a lot faster up here at high temperatures and a lot faster at low temperatures. But there's this place here where things kind of drop off at high temperatures and drop off here at low temperatures. And basically what's happening there is your enzymes just aren't working as they should. They're becoming denatured, they can't bind their substrate. So things drop off. So cold or freezing temperatures will do a ton of things that aren't so good for the cells. They'll freeze your cell membranes, they'll cause gelling, they can decrease your enzyme activity. You'll see that a lot of the times your DNA needs to pull apart for DNA replication or RNA transcription, and it won't do that just because the temperatures are too cold. And you can also get ice crystals forming that disrupt your cellular membrane at these cold temperatures. Whereas at hot temperatures, you have a cell membrane that will just break apart when the water starts to boil. Again, your enzymes will decrease in their activity, and your DNA will pull apart because the temperatures are just too cold for or too warm for the bonds to hold it together. And microbial life needs some form of liquid water. So if you're living at -15, you have to have a way to keep the water liquid. And if you're living at plus 120, you have to find a way for the water not to boil off. So here you can see, and these slides are really bad. I don't know what happened there. The psychrophiles and the thermophiles. So with the psychrophiles, they have a lot of different ways to keep their membranes intact. And one of the ways they can do this is increase the amount of unsaturated fatty acids. And when acids are saturated, fatty acids, they basically are straight. There's very strong order to them. But when they're unsaturated, they get these kinks and twists and turns and bends, and this helps maintain fluidity. So you're not just freezing up. And the same is true of the double bonds. It allows kinks and twists and bends. whereas the thermophiles have a different way of keeping their membrane intact at high temperatures. And they have special waxy chemicals that aren't really the double layer bond you'd expect to see. And they actually have, this is a normal membrane where you have your lipid bilayer, but a lot of hypothermophiles just kind of bind them together. So there's these bonds that are not as easily broken as here, and the membrane stays intact. So you also have to maintain enzyme activity, because that's huge, right? You got to carry out your metabolism at these super high or super low temperatures. And you can basically make your proteins do that in a number of ways. So in cold temperatures, everything's really fluid, and it has high catalytic efficiency. And what I mean by that is it's not binding and grabbing and staying there. It's binding, and then it's letting go, and then it's binding, and then it's letting go. So it can pump through a lot more substrate. You can also have more polar, less hydrophobic amino acids, and this just helps, again, to keep things fluid. Whereas in thermophiles, you don't want to keep things fluid. You want to keep things as structured as possible so it's not melting and pulling apart. And to do this, you can pack your proteins into a dense core and just not let water in, and then they can't boil you apart. You can add these disulfide bonds, which are down here, and again, it's just another layer of structure. And you can have charged residues like this that will form hydrogen bonds to keep things just more structured. You also have to keep your DNA together, and that's for hot, and you have to let it pull apart for cold. So psychrophiles, where things don't want to pull apart, you see a lot of AT bonds. And like we learned earlier, AT forms 2 hydrogen bonds, where GC forms 3. So we're just using more of the less hydrogen bond formers. Whereas in thermophiles, you kind of do the opposite. And you can also you can make a lot of this DNA gyrase that twists your DNA and turns it and keeps it together from pulling apart. But there's still a lot we don't understand about psychrophiles, thermophiles, extremophiles in general. And this is a picture from our lab. This is the bug that grows at -15. And you can see it doesn't look like a normal cell. Normal cells are nice and smooth on the outside. This guy's lumpy. It's got these weird things through here. And is this some kind of adaptation that's allowing it to maintain liquid water possibly on the outside? Or is it some adaptation that's protecting it against something either salt or cold? And the answer is we just don't know. So this could be a whole new adaptation to the cold. And
so what are we looking at? Like, those are 3 cells?
These are 3 cells. 1, 2, 3. And this is Planococcus halocryophilus. And you'll hear more about that this afternoon. And I'm going to end with a quote that's not really fitting on the screen. But this is a former prof of mine. It's a little crass, so I'm not going to give it to quoting a certain person. But he said, in any effed up environment on Earth, you'll find something living somewhere that's found something to eat and somehow survives. And it is crass, but it's a beautiful quote because it's true. And I was saying last night that we could almost have a drinking game at this conference where every time someone says, life will find a way, we all take a shot. And at the end of the conference, it would just be the geologist standing, I'm sure. But at the same time, it's a really good message. So in a couple years, maybe within my lifetime, if we don't find life on another planet, I think it'll mean more to me than if we do, because I can't see how it wouldn't be on another planet somewhere out there. And if we don't find it, I might have to rethink some things. So, thank you.
Anybody have questions for Jen?
So going back to those things, so at the same size with the same... about those cells, so what would a normal one look like?
Normal cells are just smooth. You don't see the bumpiness or those lines that look like spaghetti on the cell surface membrane. They're just smooth.
Okay. Yeah. Can you just you used on several slides, and when you talked, you said survive and sometimes thrive. Is there a difference? I mean, for the psychrophiles, yeah. Do you have a definition for surviving?
Hard and fast definitions, I think, are a little hard. But for example, surviving and thriving, the cells I showed you, they I would say, thrive, where generation times are maybe a couple minutes to a couple hours at room temperature with not a lot of salt. Whereas you drop them to 15, they survive, they make more cells, but it's a lot slower, and the cells seem to be more fragile. And I imagine that's kind of the way those cells would interact with nature. Things would go very slowly, but they are alive, as opposed to in the lab when we give them an all-you-can-eat buffet and warm them up, they just go to town. Would be thriving.
So I actually have a question that kind of goes to both of you. Since you introduced metabolisms and you introduced LUCA, I'm kind of curious, do we have any ideas about early ecospace, as in, are these things only doing operating on one metabolic pathway? Are they taking advantage of anything that's free? Have they differentiated yet into methanogens, methanotrophs? Do we have any even clues?
Yes, but I haven't read those papers. I know there's people that look into stuff like that for sure. I just don't know.
So when we're thinking about LUCA, I mean, are we only thinking about it being trapped to one locality, or do we think there's ecospaces everywhere?
I would say with LUCA, I would say with LUCA, you're probably going to be geographically pretty close because stuff like RNA isn't really stable. So if it's going to be transferring back and forth, it can't go very far. So I would say species differentiation. At that point, you've got geographic barriers and then lots of different spaces and stuff like that. But at least for everything up to LUCA and LUCA, probably had to be pretty close, at least in my vision.
I'd agree with that. You're talking about a lot different environment too, a lot harsher than what we have now, so it's probably.
Any other questions for Jen? All right, let's thank her again.